Hello everyone, I'm Dr. DP Bora. I welcome you all to my channel, the Microbiology Digest. So today we will be discussing another important topic in microbiology, that is stains and staining techniques. So staining procedure is one of the important prerequisite in any microbiological work. So why it is so? Because bacteria consist of clear protoplasmic matter, making them difficult to see under microscope directly. In order to visualize them or to study their structure, shape, morphology, and any other characteristics, it becomes very much necessary to make them uh, more easily visible under microscope. So this can be achieved by applying some stains which gives a contrasting color to the organism so that it can be differentiated from the background. So this is that is why staining is a primary requisite for recognition of bacteria and any uh, microbiological procedures whatever we want to perform. So staining is a prerequisite. So it may be different types depending on the requirement and the purpose of for which we are conducting our experiment. So it may be either simple staining or differential staining. Now, what is a stain? Stain is a chemical compound having one chromophore group and one oxochrome group. Both these groups are attached to a benzene ring. So here the chromophore group, it is responsible for imparting color to the organism, and the oxochrome group, it does not impart any color, but it is required in the stain. So because it is the group which is responsible for stabilization as well as other properties of the stain. So example of chromophore group includes azo groups, carbonyl groups, nitroso groups. These are chromophore groups which imparts color to the organism when it is present in a stain. Now, two terms are very much confusing. One is stain, another one is dye. So both are almost similar, but stain is a coloring agent which is for general purpose. But when it comes the term stain, it is for biological staining, biological specimen staining. We use the term stain, and dye is used for any general purpose of imparting color. Then another difference between stain and dye is that dye is a crude form of the compound, but stain is a purified form. So stains are, as I said, these are commonly used for biological specimen staining, while the dyes are mostly used for coloring textiles or other substances. Then before going to the staining procedure, so we need to have a bacteriological smear. In microbiological experiments, it is required that a smear is to be prepared to proceed for the staining procedure. So what is a smear? It is a thin film of uh, the material or the microorganism which is spread over uniformly over a surface of a glass slide. This film is dried and it is called as smear. Means smear is a thin film prepared from the materials containing the microorganisms which is prepared on a surface in a glass slide. Now, why staining is required in microbiology? So first thing is that As we have already discussed, microbes cannot be visualized because they are colorless. So we'll have to create a contrast with the background to make them visualize under the microscope. So what are the advantages of staining? It provides the contrast between the microorganism and their backgrounds. So it permits the differentiation among various morphological types, their arrangement. So different uh, properties of organism can be studied. Then some stains, they permits the study of the internal structures of a bacterial cell. Suppose like cell wall, vacuoles, nuclear bodies and other cellular organelles. So they can be visualized by applying specific stains. Then we can, after applying stain, we can go for increasing the magnification. We can observe under lower magnification or we can observe under higher magnifications. So these are the advantages when we go for staining a bacteriological smear. Now, what are the different types of stain? So, depending on the charge carried by the stain, so stains may be of three types. Number one is acidic stain, so which carries a negative charge. For example, eosin, malachite green. So, these are the acidic stains carrying a negative charge. Second type of stain is the basic stain. So these stains, they carry a positive charge. For example, methylene blue, crystal violet, safranin. These are the examples of basic stain. Then third type is the neutral stain. So they carries both positive and negative charge. Example, Giemsa stain, Leishman stain. So these neutral stains, they can impart different colors to different components of an organism. So normally, basic stains are used for staining the negatively charged molecules of bacteria. Acid stains are used for positively charged molecules. And bacterial surface, as we all know that this is slightly negatively charged. So to stain a bacteria, so we generally use basic stains for getting them visualized under microscope. Now different methods. So methods of staining, there are different methods depending on the uh, number of stains used or purpose. So first method of staining is the positive staining. So in positive staining, the bacterial cells are colored. When we apply the stain will be taken up by the, or color will be taken up by the bacterial cell, and the background will remain as a clear background. So means in positive staining, the organisms are taking the color, but backgrounds will remain clear. This is positive staining. So positive staining may be of different types. Number one, simple stain, then differential staining, then special staining. So simple staining, we use only one color or one dye. So here it provides the contrast, and the entire organism, all the organisms present in that smear, they will take up the same color because we are applying only one color in the simple staining method. So examples, Doppler's methylene blue, polychrome methylene blue, these are examples of simple staining. So what happens, the organisms, they will be uniformly taking up the same color or the color of the stain, and they can be visualized against the clear background. Next is the differential staining method. So here more than one type of dyes are used. So it can impart color to different organisms. So based on this, differentiation of bacteria are possible. So examples, gram staining method, then acid fast staining method. These are the examples of differential staining methods. Third category is the special staining. So special staining methods, here also more than one type of dye is used, and These staining methods are specifically applied for visualization of some special structures. For example, capsule. Some bacteria possess capsule. If we want to see the capsule, so we'll have to go for capsular staining method. Then spore staining techniques, these are different and they comes under special staining methods. The second type of staining, first way of staining is the positive staining. So under positive staining, we have three types: simple stain, differential staining, and special staining. So second type of staining is the negative staining. So what happens in negative staining? So here the bacterial cells, they do not take any color. Then what happens? The background is colored. Background is taking up the color. So when negative staining is applied, so organisms can be visualized as a clear uh, organisms against the colored background. So this is negative staining. So what is the difference between positive staining and negative staining? So in positive staining, the organisms will take up the color, but in negative staining, the background will take up the color. So examples of negative staining is the Indian ink, nigrosin, etc. These are negative stains. So we'll discuss one by one. First is the simple staining. So simple staining can be used to determine the cell size, cell shape, and their arrangement. So bacterial cells, they may be of different shape, different size, and they may be arranged in different forms. So these properties can be studied by applying a simple staining method. So as its name applies, only it is a very simple technique and only one type of stain is used. So example already I have cited earlier: methylene blue, safranin, crystal violet. These are some examples of positive stain, simple stain. So what is the procedure? You will have to prepare a smear, then fix it, cover the smear with the stain, then wait for 30 seconds to 1 minute. After washing, air dry, then you can observe under microscope. So if you observe under microscope, so you will see uniformly all the organisms are taking up the same color, but their arrangement, shape, and size can be studied by application of simple staining method. So if we take the example of arrangements, so bacteria, let us suppose it is a cocci, then cocci may be arranged in different forms. If it is arranged in an irregular cluster, so it is called as staphylococcus. Then if it is arranged in a group of 4 or tetrads, it may be micrococci. Or if it is arranged in a chain or pairs, it is called as streptococcus. Next is the differential staining method. So under differential staining, uh, first method is the gram staining method. So this method was developed by Danish physician Christian Gram in the year 1884. So this staining method, it is dependent on the permeability of the bacterial cell wall to the stain. So it has four components. First component is the primary stain, which is the crystal violet here. Then second is the mordant. Gram iodine is the mordant used in gram staining technique. Then third component is the decolorizer. It is the alcohol. Then fourth one is the counter stain. It is the safranin. So from the components you can see we are using here two different dyes. First one is the crystal violet, which is called as primary stain, and the second stain, which is safranin, it is called as counter stain. So gram staining, as it comes under differential staining, so after gram staining, so bacteria can be divided distinctly into two different groups. One group is called as gram positive, another group is called as gram negative. So after staining is complete, when we will be examining the smear under microscope, if it is gram positive, the gram positive organisms, they will be taking up the primary stain color and appears as purple or violet. Then if it is a gram negative, they will be taking up the counter stain color, or they will appear as red. Now, what is the principle of gram staining? So, as I said, this is dependent on the permeability of the bacterial cell wall. Now, what happens in gram-positive bacteria? The cell wall structure is somewhat different in comparison to gram-negative bacteria. In gram-negative bacteria, they have a higher amount of uh, lipid materials, and in case of gram-positive bacteria, the peptidoglycan layer, which is the major component of bacterial cell wall, it is a bit thick layer, while it is in case of gram-negative, it is a thin layer. So, because of this, once the stain enters to a bacterial cell, in case of gram positive, the diiodine complex which is formed during the staining reaction, it cannot be removed during the decolorization process. Okay, so that is why gram positive organisms, they retain the primary stain color, and in case of gram negative, because of the thin uh, as well as the higher permeability of the bacterial cell wall due to thin peptidoglycan layer, so they can be easily decolorized, and diiodine complex is removed from the organism. So in the counter staining procedure, this type of organisms, they will take up the color of the counter stain, that is safranin, and they will appear as pink in color. So this is the principle of gram staining technique. So gram positive bacteria, this is diagram you can see, photo you can see. If they are gram positive, then they will appear as uh, violet or purple, and if it is gram negative, they will appear as red or pink. Next type of differential staining method is the acid fast staining. So it is as the name says, it is a differential staining technique. Here also, organisms can be differentiated into two groups. One group is acid fast, another group is non-acid fast. So this is this technique is also again based on the cell wall structure of bacteria. And if we go back to the history of this staining method, it was first developed by Paul Ehrlich in the year 1882. Then later on, two scientists, Franz Zell and Frederick Nielsen, they modified this technique, and they made this technique more appropriate for application in microbiology. And now the modern method, it is known as Zell-Nielsen staining technique. So principle of this technique is that the acid fast organisms, some organisms are called as acid fast. For example, we can cite the example of Mycobacterium tuberculosis. So they have in the cell wall, they have a large quantity of waxy materials. This waxy material, it is called as mycolic acid. So these type of organisms, they are very difficult to stain, and once the stain is taken up by the organism, again it is very difficult to decolorize. So this waxy material, they resist the decolorization with acid alcohol. So what happens? So acid fast cells, acid fast organisms, which will show a positive reaction in acid fast staining technique. So they will be taking up the primary stain color, and the non-acid fast will take up the counter stain color. So like gram staining technique, this staining method also has four different components. First is the primary stain. It is the stain is carbolfuchsin here. Then second one is the mordant. It is heat is applied here. Then third component is the decolorizer. It is acid alcohol. Then fourth component is the counter stain, which is methylene blue here. Now materials, it is same with what are the materials required to perform this technique. It is almost same. The these staining methods, these are available commercially as a kit nowadays. So this kit, acid fast staining kit is required. So bacterial smear is required. So then one microscope, and different staining setups are required. So procedure is that first you will have to prepare a smear, then you'll have to heat fix the smear, then you'll have to cover the smear with the primary stain, that is the carboflexin, and you will have to apply heat without washing the or without removing the primary stain. So along with the stain you will have to heat till it starts steaming. So it takes generally 3 to 5 minutes. Then after that you will have to wash with water, then you will have to apply decolorizer, then again washing, then finally you will have to counter stain with methylene blue. This is the second stain, and it is applied for 1 minute. So after washing and drying, so this slide is ready for examining under microscope. So interpretation of result, it is acid fast bacteria. They will be taking up the primary stain color, they will appear as red, and non-acid fast organism, they will appear as blue due to the color of the counter stain. So in the photograph you can see it is a mixture of both acid fast and non-acid fast organisms. The blue organisms, they represent the non-acid fast type, and red or pink organisms, they represent the acid fast type of organisms. Next type of okay, before that, when we go for applying acid fast staining, so acid fast staining is the main application of this staining technique is in the diagnosis of tuberculosis and leprosy. So these organisms, they are acid fast organisms, and they are required to be stained by this technique. So some variations have also been developed. First variation already we have discussed, whatever the method, that is Zill and Nielsen method. This method is also otherwise called as hot method because we are applying the heat as a mordant here. So second variation, it is called as Kinyon method. So it is also otherwise called as cold method because we are not applying any heat as a mordant here. So next type of staining is the spatial staining. So name itself indicates these staining methods, they are for visualizing special structures of the organism. So first type of staining is the capsular staining. So some bacteria, they possess capsules which are very fragile and cannot be stained by usual method. So capsules can be stained by two ways, both positive staining and negative staining can be applied. So positive staining, examples of methods which are available are His method and Jones method. Then negative staining, example is the India ink. Then second special staining type is the endospore staining. So endospores, they are highly resistant forms of and metabolically inactive forms of some organism which are uh, found in some specific type of organisms. So when we want to visualize these endospores, so we need to apply some special staining techniques. So I have included the names only. Two methods are available. One is Hansen's method. Second one is the Schaefer-Fulton method. So these two methods are specifically applied for endospore staining technique. Then third type of stain is the flagellar stain. So as we all know, flagella is the organ of locomotion in case of bacteria. So when we want to visualize the flagella to identify whether it is a motile bacteria or non-motile, so we will have to go for specific staining technique that is called as Lifson staining technique. So Lifson staining technique is for flagellar staining. Then in some organisms we may encounter metachromatic granules. So these metachromatic granules can be stained by Albert stain, Neisser stain, Ponder stain, and Puig stain. So these four different staining methods are available for staining the metachromatic granules. So next is the staining of fungi. Fungi is another type of microorganism. So these organisms also require staining to study appropriately their shape, size, and arrangements. So for fungi, the name of the staining technique which is used is the lactophenol cotton blue staining. So this is exclusively for fungi, and this can be done by two ways. One method is called as wet mount method. Second one is called as tease mount method. Then if we want to demonstrate fungi in a uh, tissue section, so we'll have to stain with the help of periodic acid skip staining. So it is, in short, it is called as pass staining. So this is for demonstration of fungi in the tissue sections. Now in conclusion, staining techniques, as we could understand, these are very much essential tools in microbiology, because without staining we cannot study the organisms in detail. Then they helps us to identify or to study the organism or to differentiate them to specific types. Then staining also provides insights to the morphology as well as structural features of the organisms. And the special staining methods, they helps us to study the special structures whenever it is necessary. So this is what I wanted to discuss, and some review questions are always, it is important to discuss. So here important questions which may be asked from this chapter is that what are the different types of staining methods, what is simple staining, what is differential staining, then what is name some acidic stain, name some basic stain, name some neutral stain, then name mostly the special staining methods for different structures of bacteria. These are frequently asked in most of the competitive examinations. So I hope these informations are useful for you, and I have collected these informations from different sources, mostly the textbooks. And if you find that these informations are helpful, please uh, like, share, and subscribe my channel, the Microbiology Digest. It will encourage me to prepare more videos on different topics related to microbiology. So I'll definitely expect some comments from your side, so that I can further improve, or I can further include more detailed information in my subsequent videos. So with this, thank you all. Thank you everyone. Bye bye.